about this one. Just keep this one download. It's going to take some time for you. Once my demo class is completed, you're going to work on these files. Okay, you have to load this file as similar I'm going to write now. So, and you just need to send me the VCF format file, the final Excel sheet to me. Okay, so let's start this one. The Galaxy. I think everyone registered this Galaxy too. So here, as I told you in a few slides ago, last session, we need to have very high computational resources, high computational power to work with this NGS data. Right now, whatever the system we have right now, even the system what I'm working on, still it is not good for the NGS analysis. Even we cannot work with the exome sequencing data. Okay, so as I told you, we need to have very high-end server. At least we need to have a 32 to 64 gigabits of RAM, octa-core processor with 3.5 gigahertz of Intel processor we needed, and also that 12 GB of minimum of 12 GB of VGA cards we needed to do the NGS analysis. Okay, so we need to check for some alternate option. So the one of the most wonderful and best alternate option is a Galaxy. So Galaxy Web Portal, most of you is never heard about this one, or maybe some people are heard about this Galaxy Web tool. It's one of the very famous tool to do the so much of computational analysis. It is not only specific for the NGS analysis. We can do so many things, even the gene expression analysis, RNA-seq analysis, okay, even for the alignment, even for the protein modeling, okay, we can do so many things by using this use Galaxy Web Portal. Okay, so there are some the advantages I just spoke to you. The disadvantage with this use Galaxy is it has a limited space. Okay, so it will allow you for the one user for one email 200 GB of space. So previously, lucky we are now we are there. They extended the space. Previously, when I started to work with this Galaxy, at the time they gave only 50 gigabits of space. Now they extended this one. Now we have 200 gigabits of memory. We can access. We can store our information here. Okay. Okay, so for one NGS run, for one NGS analysis, approximately it's going to take 60 to 70 gigabits of space for you. If you perform the one NGS analysis run, starting from the FastQC to generation of VCF file, it's going to take 50 to 60. For example, you can see here, I run one sample. It approximately took me 54 GB of space in this Galaxy web portal. Okay, so one more advantage is with this Galaxy is if you think that your space is very limited, we can install this Galaxy in our computers. Okay, so but you have to remember this Galaxy software it's going to work only in Linux and Mac. Okay, we can install this web page locally in our computers where it is helpful for us. There is no limited space is there, and also it depends upon your storage and also the capacity of your processor the program is going to run. Okay, so program is going to run. It doesn't take there is no, even if you have a 6 GB of memory of RAM, the program is going to run, but it's going to take a lot of time for you. Even though the Galaxy, they are using the very high-end server, you can see here, I run this one sample. It took me approximately 5 to 6 hours to complete the analysis. So even for us also, when we are working with our computer, depending upon the speed of your computer, it's going to take nearly 24 hours, maybe 34 hours, 36 hours, more than that one it's going to take. So in the Galaxy platform, they are using their own high-end server. For them itself, it's going to take 4 to 5 hours to complete the NGS analysis. Okay. So this is the basics on the NGS analysis. As I told you, it is not specific for the NGS analysis. We can do so many things in the NGS analysis. Okay. Okay. Let us start this one. Let us start the session. how we can use this NGS analysis. Okay, first step, as I told you, we have to give the input file. We have to give the input file for the system. Okay, so first step, go to workflow here. Okay, so go to workflow. So first of all, we need to create a workflow. Okay, so what workflow means? So what exactly the input and what exactly the output you want. So from the first session, we know what exactly the pipeline is. As I told you here, first of all, we will get a BCL format file. From BCL2, we will generate a FastQ. From FastQ2, we are going to generate the BAM or SAM file, or BAM and SAM, sorry, SAM or BAM file. The BAM file too, we are going to generate a final version called as the VCF format file. Okay, these are the steps. We already know this one. So we have to create a pipeline. We have an option to create a pipeline in a Galaxy portal where you just give an input to the server. Automatically it will do all the process like a fast quality check. It will do the quality check, and also it will align with the reference genome. Okay, and also finally it will call the variants. So it will identify the variant, it will call the variant for you. It will do all this analysis in a single pipeline. That's the very unique thing with this Galaxy portal, that we can create our own pipeline. We can create our own flowchart, that it will run, the program will run automatically from one file to one program to other from program very easily. Okay, let us see this one. As I told you, first of all, we need to give a input. So I will show you one of the example for you. So the example flowchart I created for our analysis. Okay, you can see here. I told you here we need to give first a BCL need to convert as a FastQ. So for conversion of BCL to FastQ file, I told you we need to have a specific software that is work with the Illumina. For example, if it is the sequence is from Illumina, it need to have the Illumina based software, which is not they are not going to give a free for academically. It will come with the machine itself. So nowadays they are just converting this BCL and to FastQ. So when we are requesting the raw data to them, they are finally giving the FastQC file for us, not the BCL format file. That's a good thing. Okay, so they are giving the FastQC format file for us. From the FastQC, we can start our analysis. The first step, you can stop me if you are lost anywhere. You can stop me. You can send a message to me or unmute your screen and let me know. The first step is we have to give the input. Okay, so first we need to give an input. What is the input we need to give? We need to give a FastQC format file. Okay, so here we need to create a program called as a FastQ format, and we need to check the quality of each and every parent, whether it's a good quality or bad quality. Okay, so let us see this one. Let us first of all, I'm going to go to workflow. So let us design this one. Let us design the new workflow. Okay, here create. There is a create option is available. Now we can name this one NGS Analysis. Okay, so NGS Analysis 23 free 20. Okay, so here you can type well, version 1. Okay, version 1. Save this one. Is anyone recording this video? Yes or not? Okay, good. I think you're recording. Okay, so good. So here, if you have any doubts, you can ask Haifa. She's going to share a video for you. We can just go back and how I did this one. You can do the same version, same type of steps here. So first of all, as I told you, we need to give a input. So right here, the input. Okay, click here. You can see this input here. Input. Okay, so here input data set, click here input data set. Okay, so if you want to label them, we can label them here. We are going to mention as a forward stand. Okay, so it's a forward stand. Okay, so you need not to specify the format. If you want to specify the format, you can leave it. Sorry, you can mention here like a FastQ. Okay, you can mention the FastQ or some other things. But if you keep this one as a empty, it will automatically is going to pick up for you. Okay, so now this is a forward one. Again, click here the input data set. Input data set. So here we are going to. Yes, somebody turn on the mic. Okay, so here the reverse one. Okay, so the forward and reverse one. Now we have an input. As I told you, Illumina generally they do the pair and analysis. It means from one sample you're going to get two different type of files. One is for the forward stand and another is for the reverse stand. For each and every sample you're going to get two files, forward and reverse. So what is the next step? What is the next step once we receive the sample? For once we done the FastQC? So Haifa, what is the next step? Anyone? What is the next step? Once we receive, no, not Sambam. Okay, so first we need to check the quality. Yeah, that QC. So we need to check the QC. Yes, we need to check the quality of our sample, how much they are, how accurate they are. Okay, so first next step is, so we need to check the quality. So what you need to do is we need to add an option called as a faster. Now here we are loading all the softwares. We are loading all the pipeline. Okay, once we give the input to this here, it will automatically process everything. You need not to do each and every process in a single time. Here this is the automatic pipeline where we just need to include here your data set. Automatically it will process you. Maybe you remember the second year or first year, you remember when we are working with the gene expression analysis. Okay, so always first of all we need to collect the data sets from the database. We have to submit to the R program and we need to add various commands to it to generate an Excel sheet. Okay, every time for each and every data set you have to go through all these pipelines. But here, once you created in NGS, sorry, in a Galaxy, once you created the pipeline here, once you create a pipeline, if you submit, you can use any data set. Okay, you can use any data set. You just add a data set here. Automatically, it will give you a final Excel sheet for you. That's one of the very good, very interesting, very good thing about this Galaxy platform. Okay, next thing, as you told me, so we need to understand the FastQ. We need to check the quality of the sequence. Let me charge my PC. Okay, so here. Okay, so first up, next step is FastQC. Just type FastQC. Okay, so you will find the FastQC. Now we need to understand the quality of your sequences. So we can see here. So here we have for each and every sample. I mean, each and every sample we have to check the quality. I mean, for forward separately you need to add, and also for the reverse also you need to add separately. Okay, so here we're going to link this one. If you click this link, it will show you a various option for you. Okay, so various option for you like label. What is a label? What type of annotation you just want? What is the input file? It's there. Okay, so everything you just keep everything as a default. So everything as a default. We did not change anything. Okay, so keep this one as a default. Now we need to give the output file link to this FastQ. You can see here, I just gave the link to here. It will take this output file. It will take this output file and generate all this the FastQ report for you. Okay, even for this one also you have to do the same thing. Like for the reverse one also, we cannot use the same one. We have to use one more run for this reverse one also. Okay, so now we are going to check the only report. Here you have to remember, it is going to generate only a report for you, whether the quality of your sequence is accurate or not. Okay, not going to give any other information, not going to change your data. It's going to give only the report of your sequence. Okay, so once the quality you have checked, once it is good, you can, if it is good, then you can directly go to the alignment. Okay, the next step will be the alignment. If it is not good, if you think it's the quality is it's showing some error for you, then we have to use the other option called as a trigonometric option. Okay, we have to trim the sequence. So the software we call as one as a trigonometric. Okay. So here trigonometric. Okay, so here trigonometric software. Okay, so for each and every each and every stand, you have to use the one trigonometric tools. Okay, so for forward stand is one, and this is for the reverse one. Okay, so here it also needs the FastQ. First, we will need to change some parameters here. So if we go to trigonometric software here, we have to use we have to change some of the parameter. Okay, so here, if you want, if you're working now, we are working on this pair end. Okay, so here the pair end. Now you can see here. Now we can add the forward here. Okay, so we'll keep this one. So here the reverse one. Okay, the forward reverse one. Okay, you just need to change some of the parameters here. If you click this trigonometric, so we are working on the pair end. Okay, so perform our initial Illumina clip. Okay, S here. It's a standard sequence custom file. So I'm not going to discuss each and every parameter here. Why means it's going to take a lot of time for us if you are discussing on each and every parameter of the software. So here I'm going to give some of the commands. I mean some of the standard things what you need to do when you are working with this the trigonometric. Generally, as I told you, trigonometric is not useful for you. Why means nowadays the Illumina are giving the high quality sequences for us. If you get the fret quality is more than 20, you need not to use this one. You can directly can go to the alignment tool. Okay. So once you get the report from FastQC, check that one. If the quality score is more than 20, you can directly go and go to the alignment step. If the quality is not good, then only you have to use this trigonometric approaches. Okay, so here you need to use this standard one, and this one adapter. We just need to, for example, in our case, we use the Nextra. So for our sequence, we use the Nextra paired in. Okay, so you have to know, and also you need to remember. At least you need to know what are the parameters they used when they perform the analysis. So whatever sequence I am going to use, they use the adapter sequence as an extra. Okay, you can when they are giving a output files for you, they're also going to give all this information for you, where you can use this information, where you can find this information very easily. Okay, so here the maximum mismatch count should be default should be 20, and this the read alignment should be 30, and the accurate read adapter size should be 10, and this everything is just keep this one as a default. Okay, so this one you just keep as a default. We need not to change anything here. So here, so what is the Q? Told you the Fred, the less T. This is the how to remember. If you want a very high sequence, change this one. Okay, if you just try 30, it will go only the sequence which is having a quality Fred score is more than 30. Okay, so here to keep this one. Then a few more options are there. Inside this one, you have to use voice is not clear for everyone. It's the same problem. Now it's fine. How can I expand my screen? It's already expand max. Maybe Hadia, I think having a problem PC. Okay, so up here fine, right? I think you're recording this video, right? Haifa, I think you can better do this information to them. So here, in trigonometric operation 1, 2, we need to the first window you need to use as I told you the quality score. The second window also we need to consider drop read below specific length. Okay, if it is the length is below of the 50. Okay, so if the minimum length should be, you need to reconsider that one, the 50. Then you just go for the trimming of this adapter. Okay, so everything is default. I don't want to change anything else. So that's it. So here now we prepared our trigonometric parameters. Okay, so now we prepared. This is a very these are the default things we generally use when we work when you want to trim the reads. When you want to trim the reads, which is having a very poor Q score, Q Fred score, poor Fred score. Okay. So next one is once you've done this one, again we have to check what is the quality of this one. Okay, so what is the quality of this one? So here type this one, the FastQ. FastQ. FastQC. Okay. Okay. So here, okay. So here we done. So for first pair, again you have to do the FastQC report, and the second pair. Got it? Now, now let us see this one. Okay, so now let's save this one. Okay, now up to here, we just only check the quality. Even if you want to run this command, we can do this one. Okay, so let us see how we can run up to the quality score. So here I save this one. You have to always remember you have to save this one. So go to workflow. Okay, so here go to workflow. So click on this NGS. Here first you need to clear the new history. Okay, once you created this one, you have to create a new history and label them NGS Analysis. Okay, so you have to label them. Okay. So first of all, you need to load your data. So you need to load your own data. So the files are whatever I send it to you, you can load from here. Load your own data. Okay, so choose from local file. Okay, or just try here. Maybe it's going to work for you. Paste the link, whatever the link I send it to you. Just paste the link here. No, it's not allowing. Okay, so I think you need to load from your local system. Okay, so all the FastQ format file, whatever I shared with you, you have to load here. You have to load here. Once you load this one, you're going to see the files like this. Okay, so once you load the sample, once you load your data, you will find samples like this. This is a forward strand and this is a reverse strand. Okay, this is a forward strand and reverse strand. Both I just loaded from my data set. So now, once you loaded this one, you have to work. We have to start your workflow. Okay, so we created our workflow until FastQ. Let us run this one. How it's going to run? Let us see. So there is a play option is available for you. Okay, so here you can see here, once you click on the play, it will show you input. Okay, what is the input here? So you can see I already added here the forward stand. Okay, FW2, FW and 1, and the reverse stand 2. Okay, so you need not do anything. So I mean, we already created everything here. Click on run. Now this step will give you a faster QC format file for you with the quality of what is the quality of your sequence before and after Timnomatic. So as I told you, for our sequences, we are not using this Timnomatic approaches. Why means already they are in very high quality. It's going to take some time for us before the process is started. Okay, so here you can see here all the comments, whatever we included, it's just added. First step, it's going to give you a faster QC for the two data sets. Okay, so here you can see the 4 different files for you. One is raw data and web pages. So raw data means it will show you not the graphical matter. It will show you a raw information in a text manner. It means a numerical number. It will show you information for you. This thing it's not needed for us. Okay, these two things which is not for us. We are more going to see look onto this faster QC web page. So this first we need to check the quality. Once the quality is okay, then we can go for the alignment. But as I told you, if you're not happy with the quality, you always better to go with this trigonometric approaches. If the trigonometric approach, if you cut down, again you have to check the quality of your sequence. Okay, so how whether it's a really cut that undesired region, I mean error region, it is really remove that one or not. We can again we need to verify them by using this command. Okay, so let me show you this faster QC format file. Okay, how it exactly it looks like. If you click on this view data, okay, so if you click on data, it means the I symbol is there. It means you can view this data. So you can see, I told you here, the first line, it will provide you the information about the machine barcode, flow cell number of sample. And the second one is it will provide you about the base calls. And third one is the spacer, the quality linker. Okay. The fourth one is ACS, American Coding System codes. These are the symbol of ASC. These are the standards. These are the American standard codes. Okay, so I told you here how we can calculate this American standard codes by calculating in the Fred score values. Okay, so this is a general file you will can view in the FastQC format. Okay, let us see this one. Still it is working on. It's not going to take much time for us. I think mostly 5 to 6 minutes it's going to take to complete the analysis. So if you have a software of FastQC even in your laptop, we can easily access that faster. If you type the FastQC in Google, you can install this one. It's a Java based platform. You can install this one. Once you load your sequence data, the FastQC data, automatically it will generate so many graphs for you. Any doubts up to here, especially regarding the how we can creation of the flowcharts? It's very easy. Okay, so once you are used to it, once you are used to this Galaxy platform, actually this is the most easiest tool I have ever seen when you're working with different kinds of computational different type of biological data. Okay, so as I told you, it is not specific for NGS. We can do so many things here in a Galaxy platform. Even when I'm looking at the local host, for example, if you are able to install this Galaxy in your computer, so even I install in my laptop in Linux Place One, so you can add so many softwares there. There are so many softwares are available. Apart from here, we have very limited one. So here we have very limited softwares are there installed in this Galaxy platform, the online platform. If you install this one in the offline platform, there are so many tools are there which is developed for this local host, where you can install all the software inside of your local host. You can do so many things here. So as I told you, we can do the gene expression, we can do the protein modeling, protein annotation stuff. Not only with that, we can even work with big data by using this NGS platform. So by using this Galaxy platform. Okay, so once it is processed, I will show you the next file how it looks like. Okay, so once it is processed, it is going to take some time for us. I think, as I told you, it was going to take 5 to 6 minutes. Once it is processed, it will show you a file like this. Okay, so here it's completed. It's still, you can see here, it's still the job is it's going on. Once it is done, it will show you a symbol like this for you. Okay, so symbol like this for you. First you have to click this one, click on view data. Now you can see here 11 different types of graphs it's going to generate for you. Don't worry about the microarray. Okay, so microarray is a completely different thing, Haifa. So I'm going to talk that one later. Just first understand this concept, okay? So if you want to work with the gene expression data, that's a different pipeline. The input, but input is automatic. As I told you, you can add any input there. So if you put as a automatic, it will select any data sets. Okay, so I developed one pipeline for the gene expression also. I think in coming classes when we are working, when we want to work with the gene expression, I come to discuss further one with you. Okay, so here, so once it is as done, once the FastQC has been done, it will appear like this for you. Okay, it will appear like this for you here. Just click on View Data. If you click on View Data, now we can see here it will give you information about 11 different type of graphs are there. Okay, 11 different type of graphs are there. You can see here. This is a second sample. It means it's a reverse one, reverse sequence. So you can see now the basic statistics. It will provide the information about what are the basic information is available. So you can see here the file name, file type, what is the machine we used. You can see here Illumina 1.9, total sequences, how many reads are available in this file. So see here the number and what is the length of average length of each and every read in a sequence, and what is the GC contents are there in this in the each GC contents are there in a sequence data. So here we have a 52% of GC contents are there in our sequence information. This is a basic information which is going to provided by which provided by the FastQC. The most important one is the base per base quality score. I told you here. I told you here. It's the sequence quality. The sequence quality should be more than 20. This is a Fred code. Fred score, the quality score. I told you here the sequence should be always more than 20. And position in the read. You can see the first position in the read, it is going beyond the 20, beyond the 40. Okay, you can see this. You cannot view this sequence read length line. It means it's going beyond this one. 1, 2, 3, 4. You cannot view this one. It means they are having a very, very high quality. The quality of this basis, especially 1, 2, 3, 4 basis, is very, very high. Okay, so you can see here the line, the curve, the median line, it is not drastically decreasing. It is constantly, it is there in 28 to 40. Okay, see even the last base pair, which is around 100 base pair of 100 base pair of the reads, at the position of 100 base pair of the read, still it is having a very good quality. It is more than 22 quality is there, but the mean value is slightly higher, but still I can consider this one as a high quality sequence. I don't want to change this one. I don't want to use a trigonometric approaches. As I told you, if you are if your sequence is this yellow box size falling in this region, especially less than 20 Fred score region, then only you have to use the trigonometric approaches. If you use that one, that's the trigonometric approaches, it's going to cut down that error regions and it will give you a high quality sequences for it. But now in our case, nowadays the Illumina is giving a very high quality sequences, so we need not to worry about the trigonometric approaches. Just check the quality. So how much quality they are. Okay. So next one is the next one. It will also going to give us same information for you. So average, you can see this quality distribution, average Q Fred score. What is the mean Fred score for this sequences? You can see here the mean sequence is going beyond the 40. You can see this. It's going beyond the 40. It means okay, if it is coming like this here, less than 20 average Fred quality score, then you are having some problem with your quality of your sequence. So problem is maybe it's a contamination, maybe whenever the sequence has been called by the machine, it made some mistakes, it made some errors there. That's why maybe you have a chance to decrease the Fred score. But if you see the Fred score here, it is going beyond the 40. So as I told you, if you have a Fred score is greater than 20, we consider that one as a high quality sequence. And also you can see here the position read base pair, the ATGC, the line should be always flat like this. Okay, so difference between the A and T and G is should be greater than, not greater than the 10% of any position. So you can see this line, this each and every line corresponded to one base pair. So the line, the deviation of this line should not be greater than to their corresponding line. If it is going like this, if it is going like you can see here that at start position, it is so much of shuttering is there. If shuttering is happened throughout the sequencing, it means you're going to have a problem with your uh, problem with your sequence. And also you can check the per GC content. Okay, so you can see the per GC content always show, this is the theoretical distribution of the per GC content, and this is a GC count per read. Okay, very slight minor error is there. The sum of deviations from the normal distribution represent more than 50% of the reads. So here need not to worry about it. Still, I think it's a very high quality sequences for you. If you want more information about each and every graph, okay, you can see here, just click here, FastQC, or they will provide you the whole information about the FastQC, how we can calculate the scores, how we can check the quality of your sequence. Okay, so let us see this one, whether it's completed or not. Yeah. Still it is running. It's taking some time for us. I don't know, maybe generally it's very fast. Yeah, it's done. I can see here, it's done. If you click this one, as I told you, the same, they are the similar sequence. Okay, now you can see the high quality sequences here. Okay, so now let us see the trigonometric. It's not going to change a lot. Why means we give the cutoff value of 20. So there is no base pairs are there less than 20. Okay, there is no base pair. This region is corresponding to base pair. This is the mean value. Okay, this is the mean value, and this is the average. You can see the red line. This is the average of average FRED score. Okay, so it's not going to change a lot. Okay, so any doubts regarding the FastQC? So shall we continue to next step? Okay, so just you need to understand, we are going to check the quality of your sequences. Okay, so we're going to check the quality of your sequences by using the FastQC. We are not going to change that. We are going to change the data of your sequence. We are just checking the quality. Okay, next step is, once you are done with this, once you are confirmed with the FastQC, the next step is we have to align your sequence with the reference genome. Okay, that is called as a mapping with your reference genome. So let us see this one, how we can map this one. So again, go to that workflow. Okay, so we already saved this file, these steps. Okay, click this one, you will find edit option is there. Okay, click edit. So here, edit. Okay, so I'm going to okay, so keep this one FastQC. Why means we don't want this FastQCs. Okay, so here now next step is we need to align with the your FastQC reference genome. Okay, so as I told you, if your quality is very good, then you can directly use this two files directly to align your sequence. If your quality is bad, you just made some trimming in your reads, then you have to use this option for the alignment purpose. Okay, so the software we are going to use is BWA. Okay, right, map with BWA MEM. Okay, so map with BWA MEM. Okay, see this one. Okay, so why I prefer to use this one means even this if you use this map here still we have so many options like a map with the BW with for Illumina. So this if you use this option, even if your read is more than 100 base pair, it can easily map your sequence reads with the reference genome. Okay, click here. Okay, so first thing is we have to select this one. We have to select this one. Select that one. There is a detail option will come here for you. Okay, so here built-in index, use built-in genome index. Okay, so the next one is very important one. Here we need to select the reference genome. Okay, so reference genome we need to select. So here we're going to select human genome. So here there is few options are that like a canonical and the HG19. So we need to select HG19 only. So canonical means here you will get the reference genome for only a chromosome. So chromosome only you will get information about the chromosome. But if you use HG19, even some of the missing part like a context, some of the contexts are there which is having not embedded in the chromosome. That information, the raw information also you will get from this HG19. That's why if you are more accurate, if you are working with the specific chromosomes also, you can select this canonical option. If you want to align your sequence with the specific chromosome, then you can go for this one. So here in my case, I just want to use this HG19. Okay, so our reads are paid. It is paid. Okay, so here paid. Okay, so here the standard and mean deviation. So I'm going to add 250. Okay, if you want to read more about the standard deviation and mean, you can refer to this tool. Okay, so one more thing you have to remember. If you want to know the basic principle behind each and every tool, okay, so there is a manual available for each and every tool. Okay, if you type this tool in a Google or in a Galaxy, you'll find the manual for this tool. Okay, where they will provide you what are the inputs are they had, what are the outputs they are going to get, what are the parameters we need to use for each and every data sets. They will give a information for you. Okay, so here we are going to give a standard mean deviation. This is a default one. You can use the same set as it is. Okay, so here set group information. So I don't want to set here set group information specifically for SAMP and BAM file. Okay, so auto assign is a false one. Okay, so here the read group identifier should be, click this one, set at runtime. Okay, set at runtime. Okay, set at runtime. Okay, so here when you click this one, it automatically it's going to set up at runtime. So here next one is read group. This is a very important one. This is a major file. We need to do so many. We need to include so many parameters here. So if you are failing to input, give a proper parameters here, your sequence, your reference genome is not going to align properly with. So your sequence is not going to align properly with your reference genome. Okay. So read group name. If you can provide the group name here if you want. Okay, so and also we can here auto assign and here read group identifier. So as I told you, we can select at certain runtime, or you can provide the unique sample number. So what is this unique among the sample number is? If you check the FastQC file, if you check the FastQC file, I told you this is the flow cell number and this is a machine number. Okay, this is the instrument number and this is the flow cell number. Okay, this is the instrument ID, this is the instrument ID, and this one is a flow cell ID number. You can copy this whole information from the FastQC file and paste here at a read group identifier. Okay, so here I'm going to paste this one. Even if you don't want to keep this one, you can set at runtime. Automatically, it's going to select for you. So, but as we know this one, the flow site number and the instrumentation ID, you have to get this option here. So, auto assign one, you have to click as a no. The false one, read group number. You can provide the name of the sample name, whatever you want. For example, I'm just working on some celiac patient. I'm going to put celiac one. Okay, so here, the platform we are using the Illumina, and the auto assign of this data set should be in false mode. Library name, if you want to provide the library name, you can provide this one. Otherwise, you just keep this one as a default. Illumina library. So one. Okay, so this thing you can add by default. You can just keep this as a empty. We do not want to change anything. You can produce. You can give the description here. It is not mandatory. If you want to write down some information, you can write the description here, and you can write the time when you have started this work. You can write down. For example, here I'm just trying to write 2020 month should is 03 and 23. So flow order number. As I told you, the flow site number also is there here in this FastQ format file. Okay, this is the flow site number, flow cell number. Okay, you can copy this flow cell number, and you need to remember for each and every sequence you have to use slightly different approach. Oh, okay, then that's not a flow cell number. Okay, so keep this one as a div empty, and this one is also keep the empty. We do not have to change anything. Platform unit here. This is the flow site number. Okay, so you can see here flow cell borderline has there in this one, and here keep this one as a default, and that's it. So we need not to change anything. Okay, so majorly you have to remember, first of all, you need to select this your reference genome, and you need to set the parent. Why means we are using the both the pairs forward and reverse one, and you need to change the standard deviations, maximum minimum insertion of the length of the reads, and you need to select the group SAM and BAM file. Why means we are generating the BAM file, not the SAM file. Directly we are generating the BAM file. So first, initially software convert the SAM file, and then it will convert as a you have to keep plot in this mode. That's it. Now set alignment. So now we need input from where exactly it is coming from. So as I told you, if you want to, if your the sequence is very, you can link them forward one and one. If we think that if you work with this uh, Trinomap one, you can select after the trimming of your sequence here the reverse. Okay. So you have to say this. Trigonometric will only show the code. No. Trigonometric is not going to show you a QC for you. Trigonometric, it's going to remove the basis, the fresco reads, which is having a very poor quality. Okay, so let me explain you one more time here. Okay, now just think that this is the information you. So here, until the fresco's quality of okay, this is a read basis. This is a first base. Okay, so think that here we have a read. This is a read. Okay, read from to 39. Okay, you can this is one 40, I think. 1 to 40. Okay, this is a read 1 to 40. So the first base pair, you can see this first pair, this pair, the red score is 32. 32. When we calculate in a Fred value, it means error of this base is 0.001. It means 1 in 1,000. It is very high quality, high base qualities. So if you are moving like this, similarly, if you are moving the pattern like this, you can see that drastically it is decreased. When it comes to the approximately 26th base, it is going beyond the 20. I mean, beyond the 20 base, it means the error rate is becoming very high. It means, for example, in this case, approximately it's showing the 12 Fred score, 12, 13, 10 for just think that 10 Fred score. 10 Fred score means the more than 10%, more than 10% or more than 12% of error is there, more than 10% error is there when the base has been called by the machine. Okay, so it means the quality of this sequence is not good. So what we need to do is, whatever the sequence is not good, we need to remove them. We need to remove them. Why means you cannot trust this sequence. If you trust them, if you go through with this sequence for the alignment, you always have end up with a more false to regions. To avoid this problem, how to cut down this region? This region we need to cut down. Okay, so we need to cut down. We need to cut down from here, this region. So we need to use only 1 to 23. Okay, 23 base. This region we need to on this one. We have to use only this one to. What it will do is they help you to cut down this region. Okay, cut down this region. I will give you provide 1 to 20 only. It's not to give a full length for you. Will cut down this region, it trim this region, it will trim this region, and it will give you 23 reads only. So again, we have to check whether it is exactly down that particular error region or not by using. So finally, the conclusion is the trigonometric it will remove trim the bits which is very low quality. So the parameter we need to set up, we need to give it the instruction to the trigonometric that so if it is quality is falling below the 20, just cut down the reads. Got it? Is it fine, Haifa? Yes. Yes. Okay, so it will cut down the poor rates. Yes, that's true. The low rate quality scores. If you have very, as I told you, so now we are not using the trigonometric. What means that whatever we are getting, it's a very high quality one. Okay, so one is now we set up this alignment. Okay, you can see this information. Okay, do this alignment. Okay, let's stop this one here. Why means still we have I think approximately 25 more steps are there. Maybe it's going to take so much of time. So maybe we'll continue this class tomorrow around 11 o'clock. From 11 to 1, we are going to discuss about the remaining steps. So what your assignment is, first download the Excel, download the data, upload the data, whatever I gave it to you. So upload the data into Galaxy. Okay, so run the quality check. Okay, so if you find any errors, go for the trigonometric. If you don't find any errors there, then directly go and align with the BW file. So BW file, once you've done the BW file, once you complete the run of BW file, you can see the file like this. So once you align the sequence, you can see here, it will generate a file. Like you can see the difference when you work with the FastQC format file. So where it will show you a sequence along with the quality scores. Okay, these are the quality scores. So here in this, what is additional here? You can find the chromosomal number, and also the position of where exactly the mutation is there. So most of the information you will get from here. Okay, so you'll get some additional information in the SAM or BAM file once it is aligned with your sequence. So it's going to take some time. At least it's going to take 3 to 4 hours to align with your sequence. Once you're done with the alignment of your sequence, we need to do some more modification, some more things like removal of some of the duplicate probes we need to remove. Okay, and we need to check the what is the sum base quality is there, and we need to filter down the SAM or BAM by using the different various steps, and we need to add the different type of flag and stats are there. We need to convert the BAM file to SAM file. Okay, index statistics we just need to add. Okay, next step, last step is we are going to call the variant by using the variant calling tool. Various variant calling tool is available. We are going to use the ANOVA tool or snip effect tool to narrow down, to identify the variants in our list. Okay, so your assignment is upload two data sets, identify the FASTQ. Okay, so please take a screenshot or save them in a Word or Excel sheet somewhere. Just save them and share with me. Along with that one, you have to do the alignment. So if you want to do the trigonometric also, you're welcome to do. I think, but as I told you, it's not necessary for us. If your sequence, I don't know, I didn't check that sequence, whatever I send it to you, whether the quality is good or not. You can check that one. If the quality is fail, it's not good at the end of the reads, then you can use a trigonometric method. Once you use a trigonometric method, again you have to check the quality of the reads. Once it is passed, then use the same trigonometric FASTQ reads for the further analysis to the by alignment with the reference, using the BWA. Okay, so is any doubts, any clarification you needed? Up to here. Anyone, if you have any doubts, you can ask me up to here, the FASTQ and alignment, up to FASTQ and alignment. Okay, so after alignment, tomorrow we're going to discuss about after alignment, what we can do. After alignment is a variant calling. Okay, before we are going to call the variant, we have to add. Okay, so can you unmute your microphone, Haifa? What exactly you doubt?
Yes, doctor.
Yeah.
After we perform the alignment step, the file that we will get, is it the same page you just showed us, the list of sequences?
Mm-hmm. Yeah. So I will show you here. Once you generated this file, okay. So once this alignment is done, okay, with this one, if you click this one, okay, you can save this one. You can download this file. You can view this file, but it's a very huge file. Just show me this one. Okay, if you click on view data, you can show me what exactly the output file you got it. Just take a screenshot of this one. Even you can download this one, but it's a very big one. As I told you can see here, it's 3.3 gigabytes of space is there, storage is there. It's fine, right? Okay, it should be, yeah, it is going to generate in Excel format. Okay, so if you save that one, you can open that file in Excel format. But as I told you, it's not going to, I'm not advising that one to open Excel format. Why? I mean, it's a 3.2 gigabytes of storage is there. Why means why it is having so much of information? Mean it is consists of so many duplicates also is there. So next step is we have to remove some of the duplicate probes, which is having a homologous regions in the same BWF file. So anything else? Anyone? Bayan, Abdul Rahman, Dr. Kausar, if you have any doubts, you can ask me up to here. Anyone? No? Okay, so, yeah. Okay, fine. If you don't have any doubts, I think we can continue our class tomorrow. Around 11:30, 11, okay, 11 a.m. is fine. 11 is fine for everyone, right? So why, Hadia? Okay, so what time is going to complete? Okay, let's start 11:30. Okay, 11:30 or 11:40. Okay, 11:30 is fine, right? 11:30 to 1:30. Maybe hope we are going to complete in 1:30. Today it's a very prolonged class. Maybe I hope tomorrow we are going to end the complete session. Still, we have so many commands, so many options are there, like nearly 20 or 25 more options we need to add the complete flowchart. I will show you here. So my flowchart. So here the workflow. So here, so many steps are here. We are here. Now we are here only. So we are going to get so many information. We are here only. So up to here. So we need to add so many steps here. Okay, so to generate the final VCF format file. Okay, so that's why I told you it's not easy. The analysis of NGS is the most difficult part in next generation sequencing. So now even you can check in the Macrogen, they are sequencing the whole exome for $100 only. They are charging $200 for the bioinformatics to run this and the NGS analysis. So NGS sequencing is much cheaper than the analysis. Okay, so if you have any doubts, you can ask me. If we have 2 or 3 minutes of time, if you don't have, we will stop. We'll stop here. Okay. For the first year students,